Hello everyone, in this video we are going to talk about the general or basic principle of centrifugation and its types and types of rotors. So let's start. So this is the first slide and the basic principle of centrifugation technique. The principle of the centrifugation technique is to separate the particles suspended in the liquid media under the influence of centrifugal field. Right, so what is centrifugal field? For example, a molecule is moving, or particles are moving this circular pathway in this circular pathway. So this will be the centrifugal force towards the center, always towards the center. And when this particle will move in this direction, the velocity will always be in the tangential direction to the centrifugal force, that is 90 degree from the centrifugal force. So you think this centrifugal force towards the center, which is always towards the center in a circular path, the particles are separated from the particles of the medium, right? So this is the basic principle of centrifugation. These are placed in either the tubes or bottles in a rotor in the centrifuge. Particles differing in sizes, shape, and density are separated as the sedimentation rate is different. So as you can see that the particles and medium are placed in tube-like structures or bottles in the centrifugation machine, and then they are rotated in high speed in a circular path. So when that happens, depending on the size of the particle, like small size particle and large size particle and medium size particles, depending on their sedimentation rate, size and sedimentation rate, in their differences, particles are separated. For example, these smaller particles will have a lower sedimentation rate, that they will sediment lower. It will take more time to sediment these small particles, while these large particles will sediment in a high rate, very fast. Their sedimentation would occur very fast because their mass is greater, right? So this is what centrifugation is. Now this is another slide about your introduction. So biological centrifugation is a process that uses centrifugal force to separate and purify a mixture of biological particles in a liquid medium. So it could separate all kind of biological molecules, proteins, amino acids, even nucleus and mitochondria and DNA, and every kind of biological molecules or organelles could be separated using this centrifugation technique. Now it is a key technique for isolating and analyzing cells, subcellular fractions, supramolecular complexes, and isolated macromolecules such as proteins or nucleic acids. Now the development of the first analytical ultracentrifuge was done by Sedberg in 1920s, and the technical refinement of preparative centrifugation technique by Claude and colleagues in 1940s. So analytical ultracentrifuge was discovered by Sedberg, while preparative centrifuge technique was discovered by Claude. Now today's centrifugation technique represents a critical tool for modern biochemistry and are employed in almost all invasive and subcellular studies. For analytical centrifuge is mainly concerned with the study of purified macromolecules, isolated supramolecular assemblies. Preparative centrifugation is methodology devoted to actual separation of tissues, cells, subcellular structures, membrane vesicles, and other particles of biochemical interest. So that's about that, right? Now this is a simple example or a simple designation. Here the molecule is moving in this circular pathway, like you can see. So the centrifugal force is always towards the center, while the velocity of the molecule is always tangential to the centrifugal force, that is 90 degree between these two, right? So when these particles use the centripetal force to sediment the molecules or the particles at a higher rate. By using gravitational force and the centripetal force, they increase the rate of sedimentation. That's all happens by moving in a high speed circular pathway. So we have been talking about the sedimentation rate. So here is the formula for that. The rate of sedimentation of a particle in a centrifugal force can be shown in the following pathway. So this is the dx by dt, but dx by dt is the rate at which the particle moves towards the centrifugal field. right? So this is the rate, dx by dt is the rate at which the particle moves towards the centrifugal force. Then is equals to 2 pi r square pp, that is densities of the particle, and pm, densities of the medium. So 2 r square uh, densities of the particle minus density of the medium divided by gn. G is the centrifugal force and n is the viscosity of suspending medium. So that is the formula. Now it is important to know that if pp is equals to pm, that is if the particle the density of the particle is equals to density of the medium, then what would happen? This will go 0, right? 2 pi r square into 0, this would happen, divided by gn. So when the whole equation will be equals to 0, so dx by dt will become 0. So in that case, now the particle will not move if the density of the particle and the medium are equal, right? So the particle would not move at all if the density of the particle and the medium becomes equal. Now if the densities of the particle are greater than the medium, they will move toward the bottom of the centrifuge tube, while they will remain at the top of the tube if the particles are lighter than the medium, right? So if the particles, the density of the particle is higher than the medium, they will move downward, right? So if the density is higher, particles will gather here while the medium is at the top. But if the density of the particle is lower, then the particles will gather at the top while the medium will be at the low. So let's go to the next slide. Basic principle of sedimentation. So the effect of sedimentation due to the influence of the Earth's gravitational field versus the increased rate of sedimentation in a centrifugal field is apparent. So if we use so here is an example. To give a simple but illustrative example, crude sand particles added to a bucket of water travel slowly to the bottom of the bucket by gravitation, but sediment much faster when the bucket is swung around in a circle, right? So what it is saying is that, for example, you take a bucket and you put sand inside of it, sand particles and water, right? So there is sand in the bucket and water particles, and you keep it at a place without moving it. So what would happen? Due to gravitational force, Earth's gravitational force G, the sand particles will slowly come downwards and settle at the bottom of while the water will remain upwards. But if you swing around the bucket with the sand and water at the top, if you swing around in a circular motion this bucket, so the sedimentation would occur much faster, right? So due to this centrifugal force towards the center, that's all sedimentation is. We increase the rate of sedimentation by moving the particles in a circular motion and use the centrifugal force because of it. So that's all there is to it. So similarly, biological structures exhibit a drastic increase in sedimentation when they undergo acceleration in a centrifugal field. So when biological molecules are moved in a circular path as well, and there is a centrifugal force towards the center, those particles also sediment faster. Now, when designing a centrifugation protocol, it is important to keep in mind that the more dense a biological structure it is, the faster it sediments in a centrifugal field. So it is very easy to remind that if the particles are larger, right, if those particles which are larger or more dense will sediment faster, right, while those molecules which are small or is less dense will sediment slowly. So this is the first point. The second point is the more massive a biological particle, faster it moves in centrifugal field. Now that's easy to remind as well. The bigger the particle will be, the bigger the molecule will be, the faster it will move and the faster it will sediment. That is second point. The third is the denser the biological buffer system, the slower is the particle will move in a centrifugal field. Now we had already talked about what would happen if the particle is larger or denser, but if the medium in which the particle is moving is also denser, or the biological buffer system is denser in which the molecule is moving, then the particle will move slowly in a centrifugal field. right? Because there will be frictional force. If the medium becomes denser, if the buffer system in which the particles are present becomes denser, then there will be frictional force with the molecules, so the speed of the particles moving inside will get slower. The fourth point is, the greater is the frictional coefficient is, the slower a particle will move. Again, the same point. If the biological buffer system gets denser, the frictional coefficient will increase. If the frictional coefficient increases, the slower the particle will move inside the medium. This is the next point is, the greater the centrifugal force is, the faster the particle will sediment. So if the centrifugal force towards the center increases, the higher it will get, the faster the particle will move and the faster it will sediment, right? So the faster we will rotate a particle in a circular motion, the faster it will sediment. That's all it's saying. Now the last point, the sedimentation rate of a given particle will be 0 when the density of the particle and the surrounding medium are equals. I already told about it. If Pm is equals to Pp, that is density of the particle and the density of the medium becomes equal, then the particle will not move at all. V becomes 0. So the particle will not move at all. So this is the relative RCF, that is relative centrifugal force, RCF. The usual expression of RPM, revolution per minute, gives only the centrifugation speed and does not express the centrifugal force. So RPM is the revolution per minute. So for example, we say there is 2000 RPM centrifugation. That means per minute it takes 2000 rotation, right? So 2000, that's RPM. But what is RCF? So that RCF is the relative centrifugal force. The better expression of force generated by centrifugation is RCF, which is calculated by considering the speed of rotation RPM and the radius from the center of rotation. So RCF is equals to R into RPM square, right? So for example, if R is the radius of the circle in which the particle is moving, this is R, and RPM is the relative revolution per minute, right? So if the particle is moving in this radius R, which is equals to 10 centimeter, and it is moving, the number of rotation per minute is 2000, then the RCF will be equal to 2000 into 10, that is 20,000, right? So this will become RCF, right? That's all there is to it. Now types of centrifuge. So types of centrifuge, the first is low speed centrifuge. Now most laboratories have a standard low speed centrifuge used for routine sedimentation of heavy particles. So this is the common centrifuge we use in laboratories. The small, they don't have a very high speed centrifugation machines, they are low speed, has a maximum speed of 4000 to 5000 rpm, that is revolution per minute is within the 4000 to 5000. These instruments usually operate at room temperature with no means of temperature control. So two types of rotors are used in it, fixed angle and swinging bucket. So these low speed centrifugation machine do not produce much heat while moving, right? Because their rpm is very low, so they do not produce much heat, so they don't have any kind of temperature control mechanism. And they have two kind of rotors, fixed angle or swinging bucket. It is used for sedimentation of red blood cell until the particles are tightly packed into a pellet and supernatant separate by decantation. So this kind could be used, it is an example in which low speed centrifugation machine could be used for sedimentation of the red blood cells, right? So let's see what is a fixed angle and swinging bucket rotors. Types of rotors. So fixed angle rotor, here you can see the test tubes in which the particles are kept are fixed at a slightly bent angle, right? So sedimentation, and they move in the same fashion. they are fixed at angle, the test tube are fixed at angle, and they move in the same fashion. Sedimentating particles have only short distances to travel before pelleting. Shorter run time, the most widely used rotor type. So fixed angle rotor are the most widely used rotors, and it has a shorter run time because their angles is fixed. So the particle only have to move a very low distance to form the pellet. That's about fixed angle rotor. You can see, so centrifugal force will be on this side, so the particle will be deposited in this side. As you can see, the pellet is formed on the sides of the fixed angle rotor. The pellet is on the corner side of the test tube. Now swinging bucket rotors. The swinging bucket rotor, these are free to move in any angle, in 360 angle, in any way they want, right? From 90 degree to horizontally, in this fashion they can move in any direction, the swinging bucket rotors. So longer distance, they have to travel longer distance to allow better separation. So these kind of swinging bucket rotors allows better separation because they are not fixed. So a longer distance have to be traveled by the particles, such as in density gradient centrifugation, easier to withdraw supernatant without disturbing pellet. So in this kind of swinging bucket rotors, the pellet is sedimentated somewhere at the bottom, right? Unlike this, in which the sedimentation occurs at the side of the, in this kind the pellet is sedimentated on the side of the test tube, while in this kind the pellet is sedimented at the bottom of the test tube. So that's the difference. They have to cover a larger distance for sedimentation. So there is a better separation chance, and these fixed angle rotors have a shorter time run, and that's the difference between them. Let's go to the second type, the high speed centrifuges. We have studied about the low speed, now high speed. The high speed centrifuges are used in more sophisticated biochemical applications. Higher speed and temperature control on the rotor chamber are essential. So when you use the high speed centrifugation, a temperature control is required. Temperature control is required by these machines, because when you move a particle in such high speeds, heat will be generated. Of course, if there is a machine and it's rotating in a very high speed, heat could be generated, and if the temperature is not controlled, the particles or the biomolecules we are centrifuging could be destroyed by that heat, right? So temperature control is very much required in such kind of high speed centrifuges. Now the high speed centrifuge has a maximum speed of 15,000 to 20,000 rpm. When in low speed we saw it is 4,000 to 5,000 rpm. The high speed has a maximum speed of 15,000 to 20,000 rpm. The operator of this instrument can carefully control speed and temperature, which is required for sensitive biological samples. Of course, there are three types of rotors available for high speed centrifugation, which is fixed angle, swinging bucket, vertical rotor. So vertical rotor is something like this. You have two stands, here is one test tube, here is another test tube, and they are fixed in this angle. They could not move in any direction. They are fixed at this angle and they move in the same angle, right? And so the pellet is somewhere on the side, this side of the, like this is the test tube, so pellet will be sedimentated on the side of the test tube like this. This is the pellet where the sedimentation occur. Now ultracentrifuges. The third type of centrifuges is the ultracentrifuges. This is the most sophisticated instrument. Ultracentrifuges is a maximum speed of 65,000 rpm to 100,000 tube. The intense heat is generated due to high speed, thus the spinning chamber must be refrigerated and kept at a high vacuum. So these move in so much high speed that they require temperature control, of course, along with a vacuum is required inside the chamber, moving chamber, so that the particles are not disturbed. So vacuum is also required in such cases. It is used for both preparative work and analytical work. So there are two types of ultracentrifuges, the preparative ultracentrifuge and the analytical ultracentrifuge. Let's see about them. So analytical ultracentrifuge. An analytical ultracentrifuge spins a rotor at an accurately controlled speed and temperature. The concentration distribution of the samples is determined at known times using absorbance measurement. So analytical ultracentrifuges are generally used for analytical purposes. You know how much molecule is there, for the quality, for the quantitative mechanisms, how much molecules are there, and the concentration of them, their distribution, right? And could be measured by the absorbance measurements. Now, continuously monitor the sedimentation process. Using analytical centrifuge, we can continuously monitor the sedimentation process. So, it is use of purity of macromolecules, relative molecular mass of solutes, to change in relative molecular mass of supramolecular complexes, and conformational change in protein structure. All could be visualized using these analytical ultracentrifuge and ligand binding study, right? Because we could monitor the sedimentation process, we could monitor the sedimentation process. So we this is used for to study the purity of macromolecules, to study the relative molecular mass of solutes, and ligand binding studies and conformational change of protein structures. All could be studies using this analytical ultracentrifuge. Now, the difference, right? We already studied about what is the analytical centrifuge. Um, right, so this is used for pure samples, used to precisely determine sedimentation coefficient and um, relative weight of the molecules. Beckman model is an example of centrifuge used for these purposes. While in preparative ultracentrifuges, larger sample size can be used. No optical readout, collect fractions, analyze them after the run. So less pure samples can be used. Now, generally used to separate organelles. In this analytical ultracentrifuge, we usually separate the biomolecules like proteins and measure their purities, etc. Right? But in preparative ultracentrifuges, we separate the organelles, the tissues, and molecules. Most centrifugation work done using preparative ultracentrifuges. So, types of centrifugation. The types of centrifugation: differential pelleting. The first type is differential pelleting or differential centrifugation. So it is the most common type of centrifugation employed, the differential pelleting type of centrifugation. Tissue such as the liver is homogenized at 32 degrees in a sucrose solution that contains buffer. The homogenate is then placed in a centrifuge and spun at constant centrifugal force at a constant temperature. Now, after some time, a sediment forms at the bottom of the centrifuge called pellet and an overlying solution called a supernatant. The overlying solution is then placed in another centrifuge tube, which is then rotated at higher speed in progressing steps. So, this is differential pelleting. What we do in it is that, for example, the example here is taken of the liver, homogenized liver tissues. You can take any kind of tissues, so they're homogenized, cut into the cells. Different the cells are taken, homogenized cells, and they're put into a sucrose solution, right? So sucrose solution of different densities, right? So this is also sucrose, this is also sucrose, this is also sucrose, but they are of different densities. D1, D2, different densities of sucrose solution is taken, and within it the cells are placed. And now at constant centrifugal force and constant temperature, they are rotated, right? They are rotated. Now after some time, pellet is formed at the base, right? And the supernatant is then taken to another test tube and then once again rotated at higher speed, right? So this is kind of differential pelleting. Now, density gradient centrifugation. This type of centrifugation is mainly used to purify viruses, ribosomes, membranes, etc. Sucrose density gradient is created by gently overlaying lower concentration sucrose and higher concentration in centrifugation tubes. So what we do, there is low density, there is high density sucrose, and there is D1 density of sucrose, D2 density of sucrose, D3 density of sucrose, D4 density of sucrose, D5. So D5 has the lowest density, D1 has the highest density, right, of sucrose. So these are different density of sucrose are here. So a gradient of sucrose is produced, and the macromolecules of the cells of the viruses, ribosomes are given into it, are mixed within it, and now using this density gradient centrifuges, they are rotated at high speed. So the molecules which have density equal to d3 will sediment here, and the molecules which have density equal to d5 will sediment here, and the molecules which have high density like d1 will sediment here. So in this fashion, by density gradient centrifugation, we can separate molecules of different densities through the same process, right? And then they could be separated and studied. Now this is isopycnic centrifugation. Isopycnic centrifugation. The sample is loaded into the tube with a gradient forming solution on top or below of the preformed gradient or mixed in the cell forming gradient. The solution of the biological sample and cesium salt is uniformly distributed in a centrifuge tube and rotated in an ultracentrifuge. Under the influence of centrifugal force, the cesium salts redistribute to form a density gradient from top to bottom. Particles move to point where their bion density equals part of gradient and form bands. This is to say, the sample molecules move to the region where their density equals the density of the gradient. Right? So this is also easy to understand, as in the earlier one we studied about the different gradient density of sucroses. Now here what we do, we take cesium salt, which is CSCl, cesium chloride, right? So cesium chloride are given here of different densities, and the molecules which will have density equal to this cesium chloride, they will come here. The cesium which have the molecules which have the, so there are different cesium salts, right, and differentiated according to their gradients. So the molecules which have the gradient equal to, for example, wait a second, let me clarify this. So if this is the one, so there is C1 density 1 of cesium chloride, sorry, D1 density of cesium chloride, here is D1, here is D2, here is D3, and here is D4 and D5, right? So if cesium salts are differentiated according to their densities, and the biomolecules are given into it and rotated. So the molecules which will have density equal to the D3 will sediment here and form a band. The molecules which will have density equal to D1 will sediment here and form a band. D5, density equal to D5 will sediment here and form a band. So that's what happens in isopycnic centrifugation. Thank you for watching this video. Please like and subscribe.